So I try to make it simple as possible. But we do have an agenda, and what I would like to do is to take you through some of the items here and ask the question, what are the current infectious risks in blood products? And from the topics I saw in the conference, you've already covered a bit of that. I want to introduce you into the principles of pathogen inactivation in blood components and the currently available technologies, and also talk about the efficiency of different systems to inactivate pathogens. Talk a little bit on how clinical studies are done and what clinical studies are underway or have been done. Are there any adverse events with these blood components? And what is the status of the routine clinical use at this moment? So I'd like to start off and go into some detail on current infectious risks in blood products. And if you look at this graph here, it tells you the evolution in the time interval when AIDS was really coming around and hitting people in the 1980s until the year 2005. And you can realize that in the two countries, the UK and the US, this is based on publications mainly from there, the risk of transmitting hepatitis B virus, hepatitis C virus, or HIV came down by several log steps. So 4 to 5 logs it came down. And when you have a very stringent testing using ELISA and PCR test, you can really almost completely, but not 100%, avoid transmission of these three viruses. However, you have to be very good at testing, and something else has been discovered. And people have now looked also at the bacterial contamination risk of blood products. And when you look into at least these countries nowadays, you see that there is a much higher frequency of bacterial contamination than of contamination by these viruses in blood products. And one blood product is PC, is platelet concentrate, and the other one is RCC, it's red cell concentrate or erythrocyte concentrate. So this is a bit of the status nowadays in countries like the UK or US, where very stringent testing conditions have been implemented. If you look worldwide into these are numbers from the WHO here, where an analysis has been done looking at high-income countries, middle-income countries, and low-income countries. You see that the prevalence of transfusion-transmitted infections in blood donations by income is different by about a decade between here and here, and another tenfold increase between here and there, and can be up to a couple of percent depending on the country where you are a patient and need a blood transfusion. And there are, in this analysis, more than 25 countries where they are not able to screen for either one or even more of these three viruses in blood components. So that's an issue for treating patients who need blood. There is another issue. That is the issue that even by applying the most stringent testing criteria, which could be in the case of HIV or HCV or HBV here, both an enzyme test, which is done here. You have here your infection in your blood donor, and after a period of a couple of days or 1 or 2 weeks, maybe 4 weeks, 3 weeks, you are able to capture the infection by antibody test. As you know, that's not all. You have the possibility to directly test for the virus using PCR reaction, which actually allows you a couple of days after the infection to already detect with increasing efficiency. But there always remains a window period, which is a period where we are more or less blind with the test, and that in the end gives the observed transduction rates even with very stringent testing. And that forms the platform here to talk again about pathogen inactivation in the blood components. We talked about three viruses. This is now 10-year-old mark for a number of viruses that are emerging viruses, that we say, and viruses that can spread rapidly over the globe. And a couple of them have become important for blood transfusion. One is West Nile virus, and there is tests now in parts of the world. Another one is dengue virus here in southern US or Caribbean, which is prevalent there. And we can go further and look at the last decade. And very recently, Zika virus has become relevant. As you know, dengue fever, chikungunya fever has spread more, and it's been noted to contaminate blood components. The real risk of bacterial contamination is still there, and this is literature 10 years ago. And in the areas where there's no pathogen inactivation, the risk still nowadays is in this category. So in America, with the American Red Cross, 1 in 60,000 units, platelet units, has a bacterial contamination. In Canada, it's similar number from the Netherlands or numbers from Germany. I'm coming back to that with bacteria. It's particularly a risk in platelet components. And yes, you may perceive that the numbers are higher than the ones with viruses that I showed you are tested. To take the whole thing together, this shows you the transfusion risks with for different reasons, and this is the main infectious risks. Somewhere here there can be emerging viruses. We don't know where they are. They could be rapidly spreading and then really impose risks. They could be lying rather here as well. But in this box, we are at this moment in these areas. There can be more risk, like shown here, for HIV, HCV, HPV if testing is not fully perfect, and there's a bacterial risk. Of course, there are other transfusion risks that if you are doctors, know, and they don't have anything to do with infection, and we can't cope them in this way at the moment. So I think I could show you that we do have infectious risks in blood products that we use for transfusions currently. But there have been enormous technical developments undertaken in the last 20 years to inactivate pathogens in blood, because blood, as you know, is not a sterile component, but normally whatever we use to treat a patient, we want to be sterile. And still up to now, we cannot sterilize blood because of biological reasons, but we can bring it much closer to sterility than ever before. And that comes with the advent of chemicals, and they are very potent chemicals that have an avidity to DNA. I'm going to start off today with amortosalen and the amortosalen derivative. They are psoralins, these two, and they have been used in the treatment of patients for dermatological diseases for many, many years. They've been known to be tolerated by the body and to be in the body already. I'm going to talk a little bit about riboflavin, and maybe you also know that methylene blue is a compound which for many years has been used to photoactively treat plasma components and make them free of viral contaminations. This is UV irradiation. UVA, it activates amortosalen. UVB, the riboflavin. UVC, there's only one method that uses that. Doesn't need any other component at this time. I'm going to exclude the methylene blue part because it's mainly plasma inactivation, and concentrate on the blood products, which are red cell concentrates and platelet concentrates mainly. So I'm going to start off with amortosane hydrochloride that was used by the group of Cirrus, and they brought up and developed a line that's called Intercept products. And actually we do have people here from Cirrus and Interlux as well who represent Cirrus here in Lithuania, so there's a booth outside. If you want to know any more about the technology, they'll be there for you. The basic principle that forms the basis for being efficient is that amortosalen by itself, if you add it to a blood component, diffuses into the cells, into platelets, into leukocytes, or any viral or bacteria on its own. It's freely diffusible. It intercalates by its chemical properties into the DNA helix. And if you then illuminate and use UV light, you can permanently cross-link the DNA. And you can absolutely inhibit, and it's been shown in many examples, the replication of pathogens and of course of leukocytes, of anything that has DNA in it in the further time ahead. So this has been developed for inactivating platelets and plasma products. It can fully block replication from the DNA. The products are potent, and because platelets and plasma themselves are not dependent on DNA, and you can inactivate your pathogens. A big challenge was to develop the technique towards inactivating red cells, because here you cannot irradiate with UV. UV light doesn't reach the cells, but the S303 compound is the basis for a new line of technology where, again, this is added, in this case, to red cells, to erythrocytes, and there's a free diffusion into the entire blood components, intracellular parts, extracellular parts. Again, you get intercalation, and you get an automatic chemical reaction cross-linking that takes place over a couple of minutes to hours. You get a degradation product there, and with a very similar high efficiency, you can inactivate a pathogen in those blood components. That shows you just the layout of the technical layout. So you have photoilluminators here. You have blood bag sets that, as we have normally, but there are special bags that have in this case, the amortozolin added. That's a system here for platelet concentrates, and that's one for plasma. So you inactivate one time, one unit, and you can have 2 platelet concentrates from one inactivation step, or 3 plasma components. You can remove, and you do remove, the amortozolin with a certain device here after the inactivation. And in the case of red cells, that's a little bit different. There you have to wash out the reagents after the incubation step. It's called an exchange step. And the entire thing is available commercially for those 2 parts, and at the moment in the clinical development for the red cell part, and will be available in, I guess, a couple of years, to very efficiently treat any blood component you wanted to have for patients. I'm skipping this one because it's very technical. Just to show you, I was lucky to be involved in a clinical study with Cirrus where we inactivated red cell concentrates, and this is how it looks in the practice. So you have an extra step. You have to do it overnight, but you can do it to every unit if you want. That's the most developed and available principle to do photo inactivation of blood products. I shortly also mentioned the UVB mediated process, which is done. It's called Mirasol. It's offered by a company named Terumo. Again, you can have blood products, in this case not red cells, just platelets, and you add, in this case, not amortozolin, you can add riboflavin to it, and again you do an illumination step. Again, you get a reduction of viruses, bacteria, and parasites, and some inactivation of leukocytes. And there's a cartoon showing this with the photo inactivation molecules that find their way into cells and into the DNA. And when you then illuminate the entire thing and irradiate with your UV irradiation, you get permanent cross-link by photochemical activation step. And this way you are reducing the pathogen load within the component. The third principle is the UVC. I'm very briefly mentioning it. It's just worked for platelets. It's not yet clinically developed. It physically uses the big gap that we have here in the absorption between DNA and protein at a wavelength of 216. That's very high energy. That requires that you have a blood component that you irradiate, and you have to keep it under constant agitation, and the actual inactivation is only taking place in a very narrow area under the plastic here. But it's possible that this will also be used in practice, but only for platelet concentrates. So I think this shows you that there are some principles available to do pathogen inactivation. How effective are these? And this shows you a compilation of the data from the Intercest system, the UVA system, that's the most developed here, and you can only calculate the log reductions, and it must be greater than 5 logs, greater than 4 logs, etc., because it's very difficult to add so high amounts or small amounts to the bags to really have to physically show the inactivation. So it's greater than 5 logs, and that means that generally from most regulator authorities it's a safe and efficient procedure. And that's viruses, either HIV itself or model viruses for hepatitis C or hepatitis B shown here. Very potent also for other viruses like HTLV, West Nile, chikungunya virus, dengue virus, cytomegalovirus, which is an important virus in transplantation, oncology patients, other things like influenza, adenovirus. There are a few DNA viruses where it's a little less active. Parasites, as you know, malaria is a big topic in parts of the world, very highly efficient here. Bacteria, as I told you, which are a higher threat nowadays in components. Very efficient inactivation, again by 5 or 6 logs for both gram-negative and gram-positive species. And I told you that the incidence in blood donations and components can be in the area of a cup 1 in 2,000, 1 in 5,000. I think this is a field that has been underestimated. It's also very efficient and much more efficient than irradiation in killing leukocytes. One could say it's fully efficient. And transfusion-associated graft-versus-host disease should not be possible with this, and there are very good animal models that show that. So this shows a compilation again of pathogens that are important. This is the routine pathogens in green here. Cytomegalovirus. There's a big debate whether a positive or negative test means anything. If you inactivate, you are sure, better than by irradiation, that you eliminated the active virus. If you transfuse to immunosuppressed people. This is viruses that fall under the group of the emerging viruses here, or some protozoa that actually present some problem. And they're very efficiently inactivated, as well as I showed you gram-negative, gram-positive bacteria. Very similar numbers for the system with platelet and plasma inactivation or red cell inactivation. And as I showed you, leukocytes. There are also data out for the UVB system. UVB has less energy, so one could expect it could not be quite as efficient. Still, there are numbers like 2 to 4 or 5-fold reductions for viruses like HIV, HCV, or a model virus for hepatitis B. It's 2.5-fold here. Other viruses like CMV can be either very well inactivated or, in this case, inactivated by between 2 and 3 logs for the emerging viruses. The conclusion was that about 75% of the investigated pathogens are, in this case, we say, reduced by the other system, by the mirror-cell system, which has been made available for platelets. There are data for inactivating bacteria. I already showed you the data for the amotosalen system, which is depicted here in a series of experiments with different bacteria. And very high inactivation efficiency. These are the efficiencies with the UVB-mediated system. Again, there is efficiency on a smaller log scale. So this would hopefully convince you it's possible to inactivate pathogens on a very broad basis using UV-mediated pathogen inactivation system. How was that put into clinical practice and studies? The concept is that, of course, you first of all need to show that your components are still good, and this shows you a paper from Johannes Irsch here from Cyrus Group, looking at blood plasma, and you have different coagulation factors here. I'm not enumerating them, all of them. In principle, you have either very little loss or you have maintenance of the levels and the biological activity of these products. So the system is developed in a way that there is could be some marginal effect on the product quality, but of course the cells or the plasma components have to be efficient, and that goes throughout the systems, whether you take platelets or plasma or red cells. But then the whole thing takes enormous amounts of work to put clinically into place, because you have to perform clinical studies. For this you have to apply for licenses, and you have to go through the process. And in the case of the Intercept system, this is the status at the end of 2015. We can say that both the platelets and the plasma inactivation has gone through all the phase 1 and 2 and 3 clinical studies. The regulatory reviews has been implemented or given the licenses, and is in the marketing phase or in routine clinical use. Both in the European Union and the rest of the world, and the USA here. For the red cells, the status is in the US, it's at phase 1 and 2, and now in Europe it's progressed to phase 3 studies as well, showing that you can inactivate also red cell concentrates. I'm not going to detail this, just to show you the strategy for the Intercept system, how Intercept system have been systematically evaluated. So you try them in healthy subjects and see whether you can recover them and whether they survive and whether they're tolerated. And then you have studies, and these are multi-center studies in blue and single-center studies in red, that all have been performed and successfully evaluated, and they show in patients, in this case that patients that are thrombocytopenic, that get them as a replacement, that they are effective and comparably effective to conventional components. There has been criticism, because people would say, well, you irradiate those platelets. Are they really stopping bleeding? And a single study has not really provided the answer, but a meta-analysis of 5 studies is shown here. And this is the result, the overall result. So some studies, they are also, most of the studies are close to 1, so there's not much of a difference. And when you take them in the total, the studies, now there is good, convenient evidence that they are good to stop bleeding. Something else has been achieved, something one initially didn't want or didn't look for. As you may know, platelets are usually kept for 4 or 5 days, and that's the end of the shelf life. Now, when you inactivate them, it's been shown that you can extend the shelf life to 7 days, and there's a study that has looked at the main interval until the patients needed the next transfusion with platelets that have been aged 5 to 7 days. This is important for logistics. If you have a blood bank, you want to provide your platelets in the right time and enough, and you can concentrate better on your inventory if you know that you can extend the shelf life of platelets. So that has been shown here that it works, and another work from Jean-Claude Osseler in Belgium has nicely demonstrated it's only those numbers which is important. The worst event that you have, and why this is why you give platelets to patients in thrombocytopenic patients, because they could have get bleeding in the CNS. And that shows that there is no statistical difference with either conventional platelets or 5-day storage platelets or 7-day storage platelets in an oncology setting here. So to draw that together, there's lots of data around, and very solid and good data that this is a safe blood component which is efficient, and at the same time is a pathogen inactivated product which actually reduces, and I'll show you later, the transfusion risks here. Similar things have been done for plasma. I'm not going to show you all the details, but there have been studies showing that the Intercept plasma works in coagulopathy patients and is pharmaceutically efficient. And last series would be on the red cells. Again, just demonstrating you that both in healthy blood donors and in patients. There is a system to evaluate them in the relevant clinical situations. For in the case of red cells, you have acute transfusion or you have chronic transfusion. Some have been down with the so-called first-generation system. Now the system has been improved, and the clinical studies are also being run with this system. A little last sideway, the UVB-mediated system, the Mirasol system, has been studied in another application mode where you're not preparing blood components, you're just inactivating whole blood, and that's something that is attractive in low developed countries, in African countries, where a blood transfusion is really a blood transfusion. You take blood from a donor, put it in a bag and give it to the recipient. Yeah? And here it was shown that you can add, in this case the riboflavin, you inactivate your product with this and you transfer it into a new bag and give it to the patients. It was done in sub-Saharan Africa, so in Ghana the study was done, and the end point was, could it prevent bacterial and malaria transmission by transfusion? And there are little data out for this. However, the small study has been completed. One could look at one thing. It was known that a part of the donors have malaria, and the malaria could be shown to be transfused, because in the rest of the transfused products, where you have a red dot, we have a certain load of malaria parasites. And when you treat them with the system, you still have maybe 1 or 2 patients where you transmit malaria, but it's much less than before. So it could really work. One would look also at the transfusion efficiency in this study and look whether the hemoglobin count of the patient before the transfusion or after the transfusion is elevated. You can see that the transfusion really works. They have a higher hemoglobin here. It's a little less, obviously, later with the treated compounds, but it may also be comparable. The UVC system is very early in development. I showed you the UVC system before. We have here function data. We just have one phase 1/2 trial, and that's a system that would only be available to inactivate platelets at this time. So that's the kind of concept and the strategy for clinical trials. Just a few words still on adverse events. There are hemovigilance programs, and people look at the side effects very systematically. Cirrus does it with the Intercept product, and they are collecting the data on acute transfusion reactions there. And of course, what you want to have, that these products will be better or at least as good as conventional products. And it nicely compares, because the Intercept and Cirrus do their own hemovigilance network. There's the Swiss national network and the French national network, and they have very similar rates here for acute transfusion reactions like fever or allergy that you normally have with these components. Also, the transduction or the death by transfusion of platelet components, you still see, even in France in 2012, 2 deaths, 1 death 2013. These are patients that received bacteria through normal conventional platelets. Has completely stopped in the area of France where PI was introduced some years ago. It's only in part of France. It's gone to 0. And in Switzerland, there was a switch in 2010 after a child died with bacterially contaminated platelet. And since then, again, no fatalities with Intercept-treated platelets. And that's statistically significant. So I think we can say, yes, it's an efficient system to inactivate pathogens. It's good to make clinical studies. It's in routine use. And to finish off, there are more studies, and one of the big focuses is studies in children, because in children you are most worried about what you give, and you want to, if you would have a sterile product, it would be perfect. So this is almost perfect product. It's not only efficacious and well tolerated, but you have a significant reduction in transfusion reaction as a result of 3 different trials here with Intercept platelets. This just summarizes for the Intercept product the different marketing and regulatory steps that had to be undergone, and at least for the Intercept, I have available for you a cartoon as my last slide that shows that it is commercially available and in routine use, and has been FDA approved for platelets and plasma in the US, in Russia, in parts of Western Europe. Here in Switzerland we use it 100% for all platelet concentrates, and we are now inactivating the plasma as well. In parts of here in Arabia and in countries in Scandinavia as well, in the EU. And it's been made and increasingly being made commercially available in other parts of the world, and there is regulatory activity ongoing in other parts before it can be made commercially available. To summarize, I think we do clearly have infectious risk in blood products, both with classical virus, with emerging viruses, and with bacteria. The principles are working. We have technologies that are available, and they are efficient to inactivate pathogens, very potent with viruses and emerging viruses, but they also cover parasites, and in the case of the intercest system, very strong for bacteria. The clinical study concepts are there, studies are there. All three components are under development, or the development has already been completed in the case of intercept plasma and platelets. Adverse reactions have been reduced, specifically in the case of children, and the routine clinical use is now there with data that we could provide full bacterial protection, at least as we've done in Switzerland, decided to do, and we have less adverse reactions. With this, I'd like to thank you for your attention.